Good afternoon, ladies and gentlemen. Well, let me introduce to you the leader of the first group, our department. It is Cognitive Systems Department, and here is the leader, or the chair of the department, Lenka Vysloužilová. My name is Olga Štěpánková, and I will be talking about the BEAT department. We have decided to join both presentations because we have very close cooperation, and the main kick for deeper cooperation came from the recently awarded project CLARA, what is Horizon European project led by colleague Šivec, but one of the work packages is being conducted in our both departments, so we are trying to join our forces as much as possible. So the first department, namely the COGSYS systems, is consisting from quite a few people, as you can see here. This represents 10 full-time equivalents, so only part of the people are really in full-time job, while the others are partly cooperating or working in some hospitals or research institutes dedicated to medical domain. Namely, this is the case of the group named Neuroinformatics of Normal and Pathophysical Hearing Functions, who cooperate with the department thanks to the fact that there is a great laboratory for hearing problems, which is established in this institute. We should review all the projects on which we are working. There are 3 international projects which are being conducted in the department, namely the newly started CLARA project, plus 2 smaller projects in context of Erasmus or Interact. The last year, the department decided to somehow push the cooperation with practical life and try to transfer much more the results of previous research into something real, which can be handed out to society. That's why we are very happy that this trend is being supported by 3 edicts, which are ending this year, and we hope to be able to talk or present the results during the next meeting we will have in a year. Besides, the people at the department are working on 2 grants of the Agency for Medical Research and on 2 grants of the Technical Agency. The grants I have referred to produced quite a lot of interesting papers. The best ones are named here because they are in the top journals, and the others are interesting, but they need to. That's why we have decided to conduct or introduce in the second part of this talk to live presentations. One will be dedicated to one of the promising directions. It will be given by Hlúbik, and the other presentation will be given by our colleague who is currently in United States, and we hope he will be able to talk about our role in the CLARA project. Now, let me introduce briefly the second department, namely the Biomedical Engineering and Assistive Technologies. This department is has the review of some projects on the right side, it is important to mention that we are also cooperating in the domain of Clara, and besides we have some other research which is dedicated to AI utilization as a reinforcement tool for learning and improvement of learning results of university, which was dedicated or gained from technical agency. Here you can see the list of people. We are just 4 people working in the department, but we have some PhD students which are cooperating with us. And here we have decided to invite for the presentation here 2 people who are co-authors of very important papers which appeared in D1 and Q1 journals. So it will be Němý and Michal Huptych. I'd like to mention that Němý is currently cooperating or starting cooperation in a grant agency with the military hospital, and the first result of in the direction which will be given to this project or just this grant will be presented here. It is worth of mentioning that it is a sort of another type of transfer of results from one domain to the other, because here he is applying some basic ideas which have been used also in his PhD thesis, and for which he gained a prize from the Alzheimer Research Fund in the last year. So let me pass the floor to my colleagues, and the first one will be Milan.
All right, good morning, good afternoon, everyone. Okay, so once again, my name is Němý, and I'm a postdoc researcher here in the BEAT group. And today I'd like to talk about the results of our research on the non-invasive description of brain tumors using MRI, magnetic resonance imaging, and this is collaboration mainly with Military University Hospital and also Karolinska Institute in Sweden. So first, to give you some background, so I'll be talking about gliomas, which are a type of brain tumors. They are very much aggressive, so there's high need to really address them. So we can characterize them into 2 different ways. One way is something called grading. Tumors can be either low grade or high grade. It really means that they are either slowly growing or very aggressive, and that's important because then we can really choose and decide on the course of the treatment and surgery, for example. So this is very nice, and we know lots about it. The problem is that to know about if the tumor is high grade or low grade, we need to do a biopsy. So we need to take a piece of brain tumor out of the brain and really put it under the microscope and then see how these cells look like. So that basically means that it's like another surgery before the main surgery. And then the newest trend is finding out about gene mutations, because these brain tumors can have different gene mutations. There's like one which is called ADH gene, which is nowadays the most, or what we know is the most important gene to really predict the growth and development of the tumor. And again, this can be decided on the brain biopsy. So we need to take out again a piece of the brain of the tumor, the actual tumor, we cannot simply tell that from like the blood analysis or cerebrospinal fluid analysis. We need really piece of brain to tell. So it's pretty invasive. It comes with problems, of course. So that there's always like needs to do this non-invasively to really tell. And we know these two grading and gene mutations are really descriptive, and they can tell us a lot about if we should operate, not operate, what the treatment should be. Yeah, but this puts heavy burden on the patient. Yeah. So for this, it's possible to use MRI, which is of course non-invasive technique. Most of the patients can be scanned. Of course, it can also be a problem in some cases, but usually this is pretty fine. We already have some techniques. We already have some techniques such as apparent diffusion coefficient model or diffusion cortices imaging, which are just some ways to look into the brain using MRI, and usually it's by looking at the diffusion of water molecules in the brain. The problem is that these methods weren't developed focused on the tumors, and so they basically look at the direction of the fibers, the brain fibers on the axons. And the problem is that the glioma cells aren't really affecting the axons. So usually when we look at these existing methods, they will tell us maybe something wrong, but maybe more in severe cases which are non-operable. So they are not really designed to do this, but it's like the only option which we have so far. So for this, we proposed a new technique which is called spherical mean technique. It's been used already in multiple sclerosis, but not here. So we use it in this case also, and we designed the whole pipeline, how to like set up the MRI machines, like for the MRI sequences, how to pre-process the imaging data, and then how to use this technique. How it works in a nutshell is that it can really model separately the axons and then the tissue which is outside the axons, so we can really see what's happening in the tumor, not in the axons which are going through the tumor, which are not affected by it. So that's really good, and what we can see in the image in the right bottom corner is actually, so on the right side, that kind of remind us really like the brain and the directions which are going there, and that's actually the axons. That's like the older technology, let's say. And on the left side, that's really something which tells about like the underlying processes. Not the axons, and that's what we need. In the paper which we published in American Journal of Neuroradiology, we showed that this actually can be used here and is superior to these standard measures. For this is just like a preliminary study where we needed to first segment the tumor from non-tumorous tissue in the images by hand. So this was done by medical experts. And then we looked and investigated the diffusion properties and had like different indices. And so what we can see in the table is that, so what's called FA, that's fractional anisotropy, that's like the older measure, and then we can see. And that's what the doctors, what they can read. They know sensitivity, specificity, AUC. That's why we showed these like very simple linear measures. So that's not very ideal. However, medical experts use this, and you can see it's definitely not ideal. Sensitivity 50%, specificity 82, AUC around the midline. So that's not very good. But then the second part of the table, these are all the measures from the spherical mean technique, and we showed that there are some which are really superior. And actually, in the paper, we showed like the whole pipeline, how to set up the whole protocol, and give really like tutorial for the doctors to use this. And we showed that it's really possible. Because that's why this is important. We kind of know this method before, but it wasn't used in this kind of cohort, and there wasn't any, like a prescription for the doctors to know like how to really go step by step and show some working prototype. Next to this, we also published this review in the Neuroscience and Biobehavior Reviews, and there's another thing, but I didn't present it here because it's just like a review paper. And I also would like to mention that for the paper I presented here last year, I was awarded Dr. Jan Bureš Award for the best scientific publication in neuroscience, and also got Junior Faculty Award from the Alzheimer's and Parkinson's disease conference. And also I'll be continuing doing this research for another 5 years, as I got a Junior Star grant from the Czech Science Foundation. Right, thank you very much.
So, good afternoon, everyone. My name is Michal Huptych, and I'm going to present our last paper based on the Prague of OHKA clinical trial, which is called the time-dependent yield of invasive versus standard resuscitation strategy. This paper is based on idea of Dr. Brian Grunow from the Department of Emergency Medicine at the University of British Columbia, and we had the opportunity to collaborate in this idea with him. So, how I said, this is based on the Prague OHKA randomized clinical trial, which is, how I said, clinical trial where the data was collected from the 2013 to 2020, and the main purpose of this trial was to test two approaches of the treatment during the cardiac arrest. So it means two approach for resuscitation, some standard approach and invasive approach where is used a specific device, Lucas, and some specific assessment. And the main important thing is the ECMO, extracorporeal membrane oxygenation, which is out-of-body blood oxygenation device, and it's very important here. The study has 256 patients, 132 in the standard and 122 in the invasive arm, and the primary outcome of the study was the good neurological outcome at the 30 and 90 days after randomization. And we know from the previous analysis that the cardiopulmonary resuscitation time or long or duration is so very important, especially for the selection which kind of approach of the resuscitation we should use, especially if we have a long cardiopulmonary resuscitation longer than 45 minutes. We know that it's significantly favored for the invasive approach of the resuscitation. And we also know that in the invasive strategy and invasive approach, there are more accurate favorable neurological outcome, especially 30-day favorable neurological outcome is significantly more accurate in the invasive strategy resuscitation. So now we have some session, and now the main purpose of this study, of this paper, was to perform a secondary analysis of the Prague OHKA data in order to demonstrate the phases of benefit and time-dependent prognosis for the invasive and standard resuscitation strategy. Here we have the first description, we can say first result. You can see two distributions or two graph with the distributions of the in the left figure of the numbers of probants or patients with a good neurological outcome at the 80 day from the randomization, and on the right picture you can see the proportion of the good neurological outcome, and both the pictures are in the 5 minutes epoch based on the duration of the resuscitation. And if we want to interpret the left figure, you can see that the blue, dark blue bar are patients or probants with exclusively conventional resuscitation. It means without ECMO, without Lucas, standard resuscitation during the cardiac arrest. And you can see that there are relatively steep decline of the numbers of the patients with the good neurological outcome, while for the probants with the invasive approach of the resuscitation, there are first phase from the, let's say, 10 minutes up to 40 minutes, where we can see some one first wave of the people with good favorable outcome, or the favorable neurological outcome, and then from the 40, probably 45 minutes up to 80 minutes, you can see the second wave of these people with the good neurological outcome, but these ones are saved by ECMO, because this is using of the ECMO what make this this result. Of course, if we can compare it in the proportion, we can say again that the conventional resuscitation is again a relatively steep decline during the first hour, but the invasive arm and the ECPR treatment are relatively stable, and for all epochs, for the whole time, there are some stable band between the 11 and 35% of probants with a good neurological outcome. This is a very good result, very good promising result, but it's an approach which is called intention to treat. It means it's according to the randomization in the study during the according to the protocol. And there are problems that in the clinical research we have some problems with the crossovers. And sometimes it's necessary to switch from one arm to the second arm and so on. And it's not clear how we want. Therefore, we prepared some second view, where we use very simple calculation, where for each minute interval we just use these two equations. In the numerator is number of pulseless probants or pulseless patients with good neurological outcome at given minute, and in the denominator is total number of pulseless probants at the given minute. And if we prepare this and create the graph based on this calculation, we obtain this description, and it's very powerful, because here you can see the power of the invasive from the invasive approach of the resuscitation immediately. And of course, you can see here very clearly the second window of opportunity to save the life of the patient with the cardiac arrest, while the standard approach is almost reached the 0% of life or patient with a good neurological outcome. So thank you for your attention. If you have any question, please ask.
Thank you very much. Good afternoon, ladies and gentlemen. I would like to present our ongoing study to assess hydration in geriatric patients by bioimpedance, namely BIS, bioimpedance spectroscopy. So basically, what is our motivation? Our motivation is the fact that the hydration plays a very important role in NIH. And basically, dehydration is the most common cause of fluid electrolyte imbalance in older patients, and also the major problem in hospital admissions with these elderly people or elderly patients. The problem is that the dehydration is a complex phenomenon with no exact simple definition, which is the problem because the standard characteristic is a plasma urea-creatinine ratio that is derived from blood samples that have to be measured or taken by educated nurse, and then those samples have to be delivered to laboratory and assessed. So it takes a lot of time, it has to be done by educated personnel, and also it's invasive. Overall, there is no light and handy method for estimation of a human hydration. So what we are trying to do is to use BIS electrical parameters of human body to assess hydration, and mainly to correlate it with plasma urea-creatinine ratio. And hopefully with those electric parameters we will be able to compare it and get hydration of the patients. So in this pilot study, it's ongoing study, and this is basically the first results. We had 13 geriatric patients from age range 71 to 92 years old. So you see the patients were quite older. And the protocol was that after taking blood samples by educated nurse, and that were sent to Unilab's laboratories, we made BIS measurements, and we were measuring all BIS raw data. So we were measuring all electrical parameters of the body on all frequencies. And we were using Bodystat 5000. Basically what we are trying to do, we are trying to compare those data with biochemical parameters. And first of all, we were using 50 kilohertz phase angle, which is most commonly used by doctors these days. And then we were also using nutrition index obtained by this. That's basically the first step for us to show it to doctors, something that they know, that is more common, and then we will be following up with the directly measured electrical parameters. So you see there is just a brief overview of obtained results, and you can see the phase angles are quite low, which is typical for elderly people or sick people. Here we can see the phase angle versus urea and phase angle versus creatinine. We were calculating correlations with creatinine and then a nutrition index with creatinine, and the correlations were 0.68 and 0.61. Basically, what we are trying to solve, we are trying to use directly measured parameters, because these days these devices that are connected with body composition are normally using calculated parameters such as total body water, intracellular water, extracellular water, and those parameters are calculated by regression equations. So, as you know, error regression equations get some biases into results, so we are trying to overcome these problems or these biases by measuring direct electric parameters, which are shown here on the next slide. These parameters are Cole diagrams, which is basically reactance versus resistance on whole frequency spectrum, which is basically direct electric parameters of body. And we hope that we will be able to use these parameters to describe electrical parameters of the body and to compare those measured parameters directly. You can also see that every Cole diagram has elevated left side, which is typical for elderly people or for old people. Also, you can see that Cole diagrams, it is for every person we measured, is basically on a different place, and this is due to the body type and body shape of the patient. So overall, in the spinal study, we were able to obtain a correlation of 0.69 between phase angle on 50 kilohertz, and as I said, we were using phase angle because it's most commonly used by doctors, and they know what they are basically what basically does it mean. Then you saw the typical left side elevated code diagrams for elderly people, which we hope that with more patients we will be able to describe briefly more. Then we were able to show some further correlations with nutrition indexes, and as I said, it's an ongoing research, and because it's work with live patients, with elderly people, it's not always easy to get blood samples and the measurements. Basically, we are doing the research at the geriatric clinic, so it has to click, so that every patient can be measured. But till today, we were able to measure 42 more patients. So we get approximately 55 patients, and the research is still going. So hopefully, at the end of the first quarter, we will be around 60 or 62 patients, and after that, we will be able to assess it once again, and we will be able to use directly measured electrical parameters that might be able or might be better for description of hydration status of the patient, and we will be able to figure out new, very easy, non-invasive method for estimation of body composition / hydration of the elderly people, which might be very useful and helpful. Thank you very much.
Perfect. Thanks much for having me. Good afternoon in Prague to everyone. I will talk on the series of stuff that you guys will see probably few more years from me now, but I'll just pick up one paper that we presented today, and both all these things will be on using artificial intelligence and machine learning in brain implant devices that people use chronically at home, on daily basis. They got implanted and then go home and use the device for several years. So I'll show you some interesting results and data. This is my disclosures from last year. So this is our little CHIRK team that worked on this paper that I'll be talking about today. Vlad Sladký, my PhD student at CHIRK, and me. So talking about the new brain technologies, I'll just do the comprehensive, quick, brief review here. We are trying to design the next generation devices that helps with epilepsy management or other diseases like Parkinson or psychiatric diseases. This is what I'm showing today is the epilepsy use case, and that's essentially the device that's implanted in the chest of the people, tunneled the electrodes and essentially through the burr holes in the skull, plugged electrodes in the targets in the brain, deeper brain structures like temporal lobe here. And this particular devices has capability of sensing, continuous sensing, so we stream the data 24/7. We build the interface, the whole platform that streams the data out of the device from the people when they live their normal lives. We can test their memory score, we can look how many seizures they have, and we build a whole platform that integrates all the data from variable sensors, implantable devices, and so on, to aggregate it in the cloud and run the distributed cloud computing to get the results from the data and maybe find some biomarkers that help us to optimize the treatment, which will hopefully enable physicians to use better and more optimal treatment for the human patients in future next 5 years, 10 years, hopefully. We'll see how it goes. So today I will talk about one particular paper that's very interesting because the device also has capability of measuring the impedance. So we build in the system where we measure impedance every 30 seconds, or recently, or we are doing it every 15 minutes for a long period of time. Long period, I mean like months and years, where the device is implanted in patients. So it's interesting to observe the impedance changes. This paper was published in Journal of Neural Engineering, and main author was Richard Cui from our team at Mayo Clinic. So what is it? I hope you guys will like it, because this is engineering stuff, impedance, and that brings me back to undergrad study. So this is how the device looks like on the panel A, and this is on the panel B. It's the CT of co-registered electrodes. You can kind of see where the electrodes are in the brain here, in the two targets on both sides, and then this is the sketch of how the electrodes looks like in the tissue. And what we essentially measure with these contacts, which are pretty apart, it's like a 0.5 millimeter space between the contacts, so it covers a big area in the brain, essentially millions of neurons. So what we think it measures actually is the extracellular fluid changes and extracellular impedance changes during the time that you are measuring it. So it can be in the order of hours to catch the circadian rhythms of the brain in cellular fluid matrix changes, or it can be months to see if there are any long-term changes, maybe caused by seizures and stuff like that. And we did some measurements in beads. This paper also compares it to this saline measurement in a benchtop experiment, just to make sure that what we are measuring is correct. So the device is a simple device. It's not like a spectrograph or something like that, where you can swipe the frequencies from 0 to kilohertz. It only has essentially a measurement tool that ramps up the pulse in like 80 microseconds, and at the very end it measures the voltage. That's what's standardly called impedance in this area. So it's really just the real U over I ratio. And but what we did is testing, if you measure that, what does this compare to if you measure it by spectrograph? So usually it compares to measuring impedance at around 2 kilohertz. You can kind of see it here on this graph. So what is interesting is that we could measure the impedance from the implant for several days right after the implant, which was never done before in human. We got 5 subjects where we measured this, and you can kind of see this type of curve for everyone. So you see the parabolic and exponential kind of curve, where the impedance right after implant goes slightly down in first few days, 1-2 days, and then it slowly exponentially ramps up to the stable condition, stable level, and you can measure half-life of it and all parameters of this. So this is how it looks like. I just want to show you the gray stuff. You can kind of see how it oscillates. I'll look at it later. This is the daily oscillation of impedance. So on this bigger curve, you can see that there are daily cycles where the impedance changes during the sleep mostly, and during the day on pretty regular way. This is how it looks like for essentially almost all patients. This is one patient, all electrodes that we were measuring it. the device had 16 electrodes, so you can kind of see it's kind of similar curve on each electrode. So the other important thing for the stimulation therapies for brain stimulation is how the impedance and things like that changes when you start to stimulate the brain. So what we were testing in this paper was how does it work compared to saline, which is the red line, where there is no brain. So if you stimulate, you just keep getting the same impedance of saline because nothing is changing. But in the brain, if you start stimulating, the impedances goes down, and if you stop stimulating, the impedances recovers back within like 7 days here. So sorry, 7 hours here. And if you start stimulating, it goes again back down. So and you see kind of magnitude, order of magnitude in which it changes, and you like to see that and know that when you're starting to treat the biomarkers and find the biomarkers from brain diseases, how these biomarkers can just change based on how the impedances of sensing electrodes are changing when you stimulate. So this is an important thing, and this is how it looks like if you start stimulating at the very beginning after implant. Then about like 100 days, 150 days, the whole tissue makes long-term changes, and then it essentially settles on the same value of impedance, and from like 200 to 60 days, it won't change by the stimulation itself anymore. And so there are some long-term changes in the brain when you start stimulate, which essentially copies the days for the big studies with the stimulation devices that are showing the effectivity and outcomes of these devices for epilepsy that are measured usually, after half a year from the implant, because before that you see the fluctuation of outcomes really. So the brain really learns something in here based on how you stimulate. And the other thing that we show in this paper is based on Philip Miewald's paper that was published last year, 2 years ago, where he showed the cycles of impedance changes during sleep. Here you can see the hypnogram, and you can see that people during the day, impedance essentially grows up, and then during sleep, it recovers back down, especially during non-REM sleep. So you can kind of see the fluctuation between non-REM and REM, dreaming sleep phases, where the impedances goes back up a little bit and down, and through the night it recovers down to 0, and then the other day continues back up. So it builds up, like the sleep pleasure builds up. So in this paper with Richard, we were checking how these cycles are stable over a long period of time, like 2 years essentially, or a long time, and you can kind of see that they are stable all the time. They oscillate within these amplitude changes, but the oscillation doesn't go anywhere different. It kind of stays in the same level. And this is the thalamic target. This is the hippocampal amygdala target. So it's even in different structures of the brain. So what to take from this? It was an interesting study, which was never done before in human. Measure the impedance long time like this, and check how it can influences the therapy and the sensing capabilities of finding biomarkers and optimizing therapy. And what we found is that the minimum value is essentially after implant reaching in 2 days, which is related to scarring, to bleeding, and then it starts scarring of the tissue around the amplitude and around the electrode, and the impedance is going back up in essentially like 2 weeks or 2-3 weeks, and then it stays, when the tissue is scarred and recovered around the electrode, and then it stays kind of stable. But then it decreases also with the impedance changes, with the stimulation and then we saw the phase and the amplitude of the oscillations. The circadian rhythm of oscillations are stable over hundreds of days. So these are like main takeaways. And also this is all stable in different anatomical locations, whether you are in thalamus or hippocampus or post-hippocampal targets. So this is very useful for delivering the stimulation, finding and optimizing the stimulation for next neuromodulation devices that will enable sensing, for example. We share everything. You can find most of the things you can find on these two links here if you are interested to look up more. So this is it. Thanks for attention, and
Thank you very
I'm
much.
open to questions if there are any questions.